So good morning, good afternoon, or good evening, wherever you are. My name is Gustavo Escola, and I'm the Research and Funding Specialist at the Canadian Stem Cell Network. While we meet today on a virtual platform, I would like to begin by acknowledging the Indigenous peoples of all the lands that we're on today. The work of Canada's Stem Cell Network occurs from coast to coast and occurs on traditional, ancestral, and unceded territory of many Indigenous peoples and nations, including the Anishinaabe-Algonquin Nation, where SCN is headquartered. SCN honors, recognizes, and respects these nations as the traditional stewards of the lands and waters where we work and meet. I am very pleased to welcome everyone to our fourth and final session of the Global Regenerative Medicine Trainee Speaker Series, hosted in partnership with the Australasian Society for Stem Cell Research, the German Stem Cell Network, the Japanese Society for Regenerative Medicine, the Belgian Society for Stem Cell Research, and the Korean Society for Stem Cell Research. We would like to thank a dedicated committee of trainees drawn from each country for helping to organize these sessions and select speakers. I encourage everyone to check out the training section of SCN's website and also our home countries network or society webpage to learn about other training opportunities. I just have a few housekeeping notes before I pass it over to our trainee committee members to facilitate the session. The session will be recorded and posted publicly on SCN's YouTube channel. Each speaker will have up to 15 minutes to present and then a few minutes for questions. We encourage you to put your questions in the Q&A chat box located at the bottom of your screen. Your participation in the discussion is highly valued part of the presentation experience for the trainees, so please feel free to engage. Closed captioning is also enabled for the session. You can choose to show or hide automated closed captioning. So with that, I'll hand over to our first trainee committee representative, who is from the German Stem Cell Network, to introduce our first speaker. Gülce Parsin, over to you.
So, thank you, Gustavo. My name is Gülce Parsin, and I'm based in Germany. Currently, where are you all joining us from today? Please let us know in the chat. I am delighted to welcome our first speaker on behalf of the German Stem Cell Network, Julia Fröbel. She is currently a postdoc in Leibniz-Institute on Aging, Fritz-Lipmann-Institute from Germany, and she will be speaking about her work on the response of the murine niche to donor human hematopoietic stem cells in the NSCW41. Julia, please go ahead and begin your presentation.
Thanks, Gülce. So I'm sharing the screen now. Hopefully it works. Which one? You see it, okay. And starting the presentation now. So first of all, while you all see my black screen probably at the moment, thanks to the organizers for inviting me to this meeting here and giving me the chance to show the data of our project. As Gülce already said, we are working on the response of the murine niche to the transplantation of human hematopoietic stem cells in our mouse models. And for everyone who might not be that much into xenotransplantation models and human immune system mouse models, I quickly want to start with an introduction to this project. So when you transplant human HSCs into a mouse, you face two problems. First of all, is the host immune system, of course, that would reject the HSCs, but this has been overcome already decades ago by the use of immunodeficient mouse models. The second problem is actually the accessibility of the niche space, as the incoming human HSC is facing a bone marrow niche that is already full of murine endogenous HSCs. So far, this was overcome by a total body irradiation of the niche, of the mice, getting rid of all the hematopoietic stem cells in this niche. But this irradiation causes so-called radiation-induced bystander effects that do not only have an effect on the niche cells that are actually staying there, but also on the incoming HSCs that we transplant them into. So what we are using here in our lab is a way to transplant human cells into mice without using irradiation. And this is done actually by a deficiency in the murine HSC KIT receptor. So the so-called W41 mutation of this KIT receptor makes their ability to keep themselves stable in the niche a bit decreased. So when we transplant human HSCs into this niche, they are basically able to kick out the murine HSCs without any further need for irradiation. So we established a few mouse models using this W41 KIT mutation in them. They are called NSG-W41. We also put another transgene for human IL-7 into this model to also be able to analyze human T cells in this mouse. And we further, very recently, established a model that is BLAK6, based on a BLAK6 background of these mice. They are called RGW41, CD47, also using the immunodeficiency genes for basically RAC2 knockout and IL-2 receptor gamma knockout, the KIT mutation, and another knockout for CD47, which basically via the axis of SIRP alpha and CD47 makes these mice macrophages unable to, they are not able to recognize foreign incoming cells. So enabling us to transplant these human HSCs there. So with these mouse models, we are now in a position to reach very high human cell levels in the peripheral blood of these mice. As you can see here, NSG reaching up to about 80%, RGW41, CD47 as well, above 60% of human cells in the peripheral blood. And how is this actually happening? So when we look at the bone marrow of these mice, we see that the number of murine HSCs in these mice changes after transplantation of human HSCs only in the non-irradiated NSG-W41 mice. Yeah, so via this inability to keep themselves in the niche due to their W41 KIT mutation, we are lowering the number of the endogenous murine HSCs in this NSG-W41 model, and this enables us to actually get a higher number of human HSCs into the bone marrow of these mice. So you can also very nicely see this on this immunofluorescence pictures where we have the human leukocytes in green in the bone marrow, and in red you see the murine leukocytes. And as you can already see, the NSG-W41 model shows way more green in the bone marrow than in the irradiated NSG. We further had a look at how these cells behave functionally in these mice. So using secondary transplantations and limited dilution assays, we were able to show that when we transplant human donor cells or like human leukocytes from the first recipients of our transplantation to a secondary recipient, we can show that in the NSG, only 1 cell out of 4 million cells is actually a functioning human hematopoietic stem cell. While in our model, in the NSG-W41, we can see that this is 1 cell out of only 800,000 cells. And this is basically enabling us to see that the functional human HSCs in the NSG-W41 model are at a tenfold higher level than in the irradiated NSG mice. So now we were wondering what happens there. Why is this happening? Why is this engraftment so stable? And why do we have continuous hematopoietic output? Is there anything that could make or show us what is responsible for this? And of course, our first view was into the niche of these cells. So in the bone marrow, the HSCs are surrounded by a lot of other cell types, having mesenchymal stromal cells here, endothelial cells, osteoprogenitors, adipocytes, megakaryocytes, macrophages, or here nerval cells. And all of them are producing a lot of different chemokines, cytokines, and other factors for our HSC to use or to cling to. So first of all, we analyzed how does these cell types change in number when we transplant human HSCs into this niche. And what you can nicely see, while we don't see any difference in endothelial cell numbers and the slight increase in osteoprogenitors, we see a very significant 5-fold increase in the number of MSCs upon transplantation of human cells into this mouse model. And what we could also show is that when we follow these mice after transplantation for like half a year of time, we see that the number of human HSCs has a peak at 15 weeks and then slowly goes down again. And we see exactly the same thing for our MSCs. So we again see a peak at 15 weeks after transplantation, and then their numbers drop again. So we were wondering, how is this important? So what is happening here? That's why we sorted the MSCs of the non-transplanted as well as transplanted mice. And what you can see here is that they actually cluster nicely together. While in blue, you see the non-transplanted, and in red, you see the humanized mice MSCs. When we then put them on a more cell type specific axis, we see that in the MSCs of the non-transplanted mice, we have kind of a mostly adipocytic and osteocytic primed MSCs. And in the humanized ones, we only see the green, basically only end up with adipocytic primed MSCs that you can also see here in the quantification. In the humanized mice, we have only 12% osteoprimed MSCs left. And we also saw this in the downregulation of the skeletal system development of these MSCs of the humanized mice. Of course, we wanted to see this on a functional basis as well, not only RNA basis. So we in vitro differentiated these MSCs and forced them to get into this osteogenic differentiation line. And what you can see here is that after transplantation, these MSCs show a way lower osteogenic potential than the ones that did not get in contact with the human transplanted cells. And further on, when we analyzed the bones of our mice via micro-CT, we also saw a lowered bone density in these mice that got transplanted with human cells. And this is actually also something that happens in the setting of human hematopoietic stem cell transplantations. As a few years ago, it was discovered that one of the long-term effects or side effects of a transplantation of human hematopoietic stem cells is actually osteoporosis. So, of course, we now wanted to use this knowledge that we gained to somehow modulate our engraftment of the MSC, of the HSCs in the niche, and used, of course, the MSCs for this. So what we now did was we have this new Black6-based mouse model that we can humanize. And first of all, of course, we compared it in terms of how it does function in comparison to our established NSGW41 model. And as you can see nicely here, we have the same amount of human HSCs that engraft into this RGW41 CD47 mice. And we also see the same response of the MSCs, while after transplantation, we see increased numbers. So now being on a Black6 background enables us to actually get a lot of Cree lines and Flox lines that we can have bought by some companies into these mice easily, because we are already on the Black6 background where most of these Cree lines are sold on. So we used a Cree line that is especially targeting the MSCs, the leptin receptor Cree, and we also got another a mouse line bred into there, which is expressing a diphtheria toxin receptor on these MSCs, and this leptin receptor Cree expressing MSCs. So when we now inject a diphtheria toxin into our mice, this enables us to actually get rid of all these MSCs that were here in the beginning. And we can now see what happens to the human HSCs when we have no influence anymore of these cells. We further established another model where we again targeted the leptin receptor-free MSCs, but this time we didn't want to get rid of the whole cell, but we just targeted the stem cell factor. So while this is gone in these mice, the stem cell factor is gone that is produced by the MSCs, we still, and this is very important actually, we still have stem cell factor produced by other cells like the osteoprogenitors, adipocytes, or endothelial cells. And we only have MSCs not producing SCF. So let's see how we were able to use these mice. So looking at the first model with the DTR, of course, before we inject DTR, we engrafted the human cells in these mice, and you see there's no difference between positive or negative cell, mice in terms of blood or chimerism. Afterwards, when we inject the DT, we went on and looked for the niche cells. So as you can see, where we don't see any difference in the osteoprogenitors or endothelial cells, we see a significantly decrease in the MSCs of these mice. And of course, we went on and looked at the human cells. So when we don't have MSCs in these bone marrow niches anymore, what is happening is that we actually also lose the human stem cells in this bone marrow. And again, when we look at the potential to form colonies, like what is defining the stem cells here, then we see that in the mice that don't have MSCs anymore, we also see a lower human colony potential. So normally when cells are lost from the bone marrow, they get mobilized. So of course we were interested in where do they go? Oh, sorry. Where do they go? And we found out that they were actually mobilized and ended up in the spleen of these mice. So we found a significantly higher level of colonies as well as of the human stem cells in the spleen of these humanized mice that did not have MSCs anymore. And our second model, we basically have so we still have the cells, but we don't have the SCF produced by these cells. And what you first see, or what we first checked, was that we have the same number of cells in these mice. So we don't see any significant difference between negative or positive cells in terms of MSC, endothelial, or osteoprogenitor numbers. But what we definitely saw directly after transplanting them with human hematopoietic stem cells is that we basically don't see, or just to a very low account, we see engraftment of human cells in these mice. And of course, at the end of this transplantation time, after 22 weeks, when we analyzed the bone marrow, we saw again the significantly downregulated or significantly decreased numbers of human leukocytes in the bone marrow, and especially also the human HSCs were in lower numbers in these mice. And this time, of course, we also saw lower HSC numbers, human HSC numbers in the spleen, because when there's nothing engrafted, of course, there cannot be anything mobilized or relocated anywhere. So to come to my conclusions, I hope I could convince you that with our KIT-deficient mice, we see a stable human engraftment by the direct replacement of the endogenous stem cells. And this is allowing us to show or to have continuous hematopoiesis in these mice. We also showed that the murine bone marrow niche interacts with these transplanted human cells by an expansion of the MSCs of the niche. And this ends up with a lower osteogenic differentiation and bone density of these bones. And with our newly established models, we showed that the murine MSCs anchor these human HSPCs in the bone marrow niche. And especially the stem cell factor produced by these MSCs is the one that the human HSCs depend on for their engraftment. So with that, I'm at the end of my talk. I want to thank all the collaborators of this work, especially Susi Ramik. She's here in the upper corner. It's a former colleague of my lab here, and she did a lot of work on this project as well. I also want to thank all my colleagues from the Jena lab here of Claudia Vasco, and of course you for your attention.
Wow, thank you, Julia, for your fantastic talk. It was extremely well done. Also, many thanks to Susan as well. So we are extremely interested in what comes next, basically. Now we have time for a few questions from the audience. You can just drop your question to the Q&A part in the chat below. And where people are typing, I actually do have a little question. And I was wondering if this interaction between murine mesenchymal stromal cells and the dependence on the human hematopoietic stem cells together with murine MSCs with the stem cell factor and KIT interaction, would this be also applicable to the human AML samples or human MDS? Could there be a communication within the murine niche with human AML samples in this case?
Yes, I think so. So there's plans actually to transplant also cells from patients. Yeah, we are not the only ones doing this, of course. Collaborators of us already did so, and I mean, so far this is only done in the NSG or in NSGW41 models. But as we now have this modular system kind of at hand, of course, we also want to do the transplantations in these and see if we can maybe even like lower the burden of the AML when we just get rid of the MSCs or basically of the one of the proteins produced by them.
Okay, thank you. Thank you again, Julia, so for taking the time today, and maybe we should move on. We especially encourage audience to contact you, of course, to keep the conversation going, basically. And now I would like to hand over to Bradley to introduce the next speaker. Thank you so much.
Thanks, Gulce. My name is Bradley Valaton, and I'm happy to welcome the next speaker on behalf of the Belgian Society for Stem Cell Research. The second speaker for the session is Paraskevi Athanasouli, also known as Vula, and Vula is currently finishing her PhD at KU Leuven here in Belgium, and she will be speaking about her work on the Wnt TCF7L1 transcriptional repressor axis drives primitive endoderm formation by antagonizing naive and formative pluripotency. Vula, please go ahead and begin your presentation.
Yes, thank you, Bradley. Okay, let me share. Okay. You see my screen?
Yes.
Okay, not like the full screen.
Yeah, just the slide.
Okay. So good afternoon. Thank you everyone for being here. I want to thank the organizers that gave me the opportunity to present my work here. Today, yes, the title of the presentation is the Wnt TCF7L1 transcriptional repressor axis drives primitive endoderm formation by antagonizing naive and formative pluripotency. So I will start with a brief introduction regarding the project. And so since our topic is mouse preimplantation development, I would like to tell you that preimplantation development in mouse proceeds from one cell zygote to the blastocyst stage, which at 4.5 implants in the uterus of the mother for future development. During this short time window, only 4.5 days, the first differentiation events take place and are, as we call them, lineage specifications. The first lineage specification separates the trophectoderm, which is the outer layer of the embryo, and the inner cell mass. Later, after implantation, the trophectoderm will give rise to the placenta. The second lineage specification resolves the inner cell mass into two populations, the epiblast and the primitive endoderm. After implantation, the epiblast will give rise to all the tissues of the embryo, and primitive endoderm will contribute to the formation of the yolk sac. So in this project, we focused on the second lineage specification, where at around day 3 of development, the inner cell mass co-express two central markers, NANOG and GATA6. And following their mutual exclusions, the cells will either become only NANOG positive, so they will become epiblast cells, or GATA6 positive and only, and they will become primitive endoderm. And we know from literature that this mutual exclusion happens because these two factors represses each other expression. Also, this another evidence for this reciprocal inhibition between them is that disruption of nanog expression in mouse embryos results in the initiation of the primitive endoderm program, and the other way around, depletion of GATA6 results in inner cell mass that only consists of nanog positive cells, so only epiblast. So even though the mutual inhibition of epiblast and primitive endoderm program is probably the basis for the establishment of these populations, we still have limited knowledge about the upstream signals. The upstream signals that drive this process. And this brings me to the aim of this project, which is to uncover the molecular mechanism that regulates the epiblast versus primitive endoderm specification. So in standard culture conditions for mouse embryonic stem cells, and this is serum leaf conditions, we can identify at least two different populations. Most of the cells, transcriptionally and functionally, resemble the preimplantation epiblast. So we call them naive epiblast, and they are nanog positive. Under differentiation conditions, these cells can differentiate towards post-implantation epiblast lineages and ultimately to all the embryonic lineages. Now, there is a small fraction of cells in culture that resemble the primitive endoderm in vivo, and they express GATA6. However, naive epiblast cells can spontaneously differentiate to primitive endoderm-like cells, either in proper culture conditions or by depleting the expression of epiblast-specific genes like nanog, master regulator of the epiblast program, or by forced expression of primitive endoderm genes like GATA6. And by this, we understand that this antagonistic relationship between them is present in vitro, as we saw also in vivo. And this then makes mouse embryonic stem cells an excellent model to study this process. So by using these conditions, we were able to identify these two populations by the presence of two surface markers, one pluripotency specific, PCAM1, and one primitive endoderm specific, PDGF receptor alpha. We sorted and we characterized these two populations, and we saw that they share a similar transcriptional signature with epiblast and primitive endoderm-like cells, and this establishing them as their in vitro counterparts, and we use them as such for the rest of the study. So to identify the signaling pathways that might be involved in this regulation of epiblast and primitive endoderm specification, we performed pathway enrichment analysis for the in vitro populations and in mouse embryos using publicly available single-cell RNA sequencing data. And in both cases, we found Wnt signaling pathway as one of the deregulated pathways between them. Also, gene expression analysis showed that naive epiblast cells in vitro and epiblast cells in embryos are characterized by high expression of Wnt positive regulators and lower expression of Wnt negative regulators. And the other way around for primitive endoderm-like cells and the primitive endoderm cells in the embryo. Indicating that primitive endoderm cells are characterized by limited Wnt signaling activity. Okay. So the effector molecule now of the Wnt signaling is beta-catenin, which when is accumulated and translocates to the nucleus, and it facilitates Wnt target gene transcription by binding to the TCF-LEF family of transcription factors. So we performed Western blot for active beta-catenin for the in vitro populations and immunofluorescence analysis for active beta-catenin in mouse blastocysts. And we demonstrate, and in both cases, we saw that primitive endoderm-like cells and primitive endoderm cells in the embryo exhibit lower levels of active beta-catenin, indicating that naive epiblast cells hold a Wnt on state, whereas the primitive endoderm cells are characterized by a Wnt off state. So then we wanted, of course, to investigate deeper the role of Wnt signaling. So we inhibited the pathway using a physiological inhibitor, DKK1. And we saw that inhibition of the pathway resulted in an increase of primitive endoderm-like cells, as well as an increase of primitive endoderm genes. Then we also treated mouse embryos with the same inhibitor, and we again observed an increase of primitive endoderm cells, so GATA6 positive cells in the inner cell mass, showing that in both cases, Wnt signaling inhibition promotes primitive endoderm specification. Now, to address whether the effect that we see is a transcriptional effect and whether it is mediated by the TCF-LEF factors, we treated the cells with ICRT3, which is a small molecule that actually inhibits the binding of beta-catenin to the TCF factors, meaning that then the TCFs can have only the repressive function. And we saw that ICRT3 treatment resulted in an increase of primitive endoderm cells and primitive endoderm markers, showing that is mainly the repressive activity of the TCF factors that is implicated in primitive endoderm specification. Now, in mouse embryonic stem cells, the most abundant TCF is TCF7L1, which is considered a transcriptional repressor. And since we had a hint from our previous experiment that the responsible factor probably is a repressor, we wanted to investigate whether TCF7L1 has a role in primitive endoderm differentiation. And depletion of TCF7L1 resulted in a significant decrease of the endogenous primitive endoderm-like cell populations, indicating that TCF7L1 is required for this cell heterogeneity. So, okay, then we radically induce primitive endoderm differentiation in vitro by using retinoic acid, and we saw that knockout cells were unable to differentiate towards primitive endoderm, as we show by FACS analysis, gene expression analysis of primitive endoderm genes, and immunofluorescence analysis for GATA6. In the other way, however, the knockout cells were able to exit pluripotency and to differentiate towards an embryonic lineage, the neuroectoderm, demonstrating that TCF7L1 depletion specifically compromises primitive endoderm specification. Also, disruption of TCF7L1 expression in mouse embryos resulted in impaired primitive endoderm formation, indicating that TCF7L1 is indispensable for correct primitive endoderm formation. To further look into the role of TCF7L1 in primitive endoderm specification, we overexpress TCF7L1 in mouse embryonic stem cells for up to 8 days. And after 48 hours of overexpression, we observed a progressive upregulation of primitive endoderm genes and downregulation of pluripotency genes. And we know that after implantation, primitive endoderm gives rise into two tissues, the visceral and the parietal endoderm. and to extensively study the role of TCF7L1 during primitive endoderm specification, we performed transcriptome analysis, and we saw that after 8 days of overexpression, primitive endoderm, parietal endoderm, and visceral endoderm genes were highly upregulated, showing that TCF7L1 expression was enough to induce primitive endoderm fate. We wanted, of course, also to study the dynamics of this process, how this happens. And that's why we studied earlier time points of the overexpression, and we found that the first event that takes place upon overexpression is the downregulation of naive and formative pluripotency, while primitive endoderm gene activation follows, indicating that probably TCF7L1 functions as a repressor of these genes, and this repression is necessary for the initiation of the primitive endoderm program. Now, formative pluripotency is an intermediate state required for the cell commitment towards the post-implantation or primed epiblast. And to assess whether TCF7L1 directly regulates formative pluripotency genes along with the regular pluripotency genes, we integrated RNA sequencing data of the day 1 downregulated genes with TCF7L1 chip sequencing data on mouse embryonic stem cells, and we found that TCF7L1 binds on those genes and represses them, leading to their down regulation. As a proof of concept, we performed differentiation towards post-implantation epiblast, and indeed we found that overexpressing cells were resistant to epiblast differentiation, confirming our hypothesis that TCF7L1 acts as a barrier. In summary, we demonstrated that Wnt inhibition induces primitive endoderm specification in vitro and in embryos, and that the key player in this process is TCF7L1, since it was indispensable for correct primitive endoderm formation. Our findings suggest that TCF7L1 binds and represses essential genes to the epiblast fate, disrupts then the biopotentiality of the cells, and enforces the cells into a primitive endoderm fate at the expense of the epiblast. And this may suggest a possible mechanism of this binary fate regulation. I want to thank my supervisor, Frederick Lewis, and the lab and all the team. And I want to thank you for your attention, and I'm happy to take your questions. Thank you.
Thanks for the talk, Vula. While waiting for questions, I guess I'll ask one of my own. So in vitro and in vivo, where does the Wnt for this come from?
Yes, in vivo, there may be two explanations. So we know that the endometrium expresses some Wnt components, and actually it also expresses DKK1. So it may come from that, or it may come, at least in mouse that we have multiple embryos, it can also be in a paracrine way, like that, because the embryo itself also produces some Wnt components. So it can be autocrine from the same embryo to the same embryo, or paracrine between embryos. And in vitro, what do you mean in vitro?
I mean, in your cell culture, I guess, are you using Wnt activators or inhibitors?
No, we don't use it. So that was the thing, that in serum leaf conditions you have these cells present, the primitive endoderm-like cells present, and we think that what maintains this heterogeneity is actually TCF7L1, and when yes, because when we knocked it out, we lost it. So we don't have Wnt modulators like in our regular culture. We used it just to modulate the pathway and see what happens.
Thanks for your questions. I think we're running out of time, so we'll move on to Freshta. Thanks. There's still questions if you want to answer online or in the chat.
Hey. Thank you, Bradley. My name is Freshta Younesi, and I'm based in Canada. I'm excited to welcome the third and the last speaker of this session on behalf of the Canadian Stem Cell Network. Our final speaker is John Edgar. Is currently postdoctoral fellow at University of British Columbia and will be speaking about their work on multi-objective optimization reveals time and dose-dependent inflammatory cytokine-mediated regulation of human stem cell-derived T-cell development. Please go ahead and begin your presentation, John.
Great, thank you for the introduction. I mean, that title is a bit of a mouthful, isn't it? So I've made a simpler title here, which I think captures the same thing that I'm going to talk about today, which is really my work to engineer an in vitro niche that kind of replicates what our bodies are normally doing to, every day to generate T cells from stem cells. So T cells are these really amazing immune cells, part of our adaptive immune system that, along with B cells, provide like memory to previous infections. There's broadly two classes of T cells. One is CD8 or cytotoxic T cells. These are cells that normally, roam through our bodies and serve a healthy and infected tissue and or cells and detect whether they've been infected by a virus or whether they're cancerous. And they do this through this important receptor called a T cell receptor. And this is what really gives them their specificity to be able to detect different pathogens or cancer. And when they detect something that's foreign or that is pathogenic, they can release a variety of cytokines such as interferon and TNF-alpha, and also other molecules such as perforin and granzyme B that effectively lyse these cells and destroy them. The other type of T cell is known as a CD4 helper T cell. And these cells interact with professional antigen presenting cells called dendritic cells that themselves are surveying antigen or peptides in circulation and to find things that in our body that are not supposed to be there, and then activate these helper T cells, which can then secrete a variety of different cytokines and immune effectors that activate CD8 cytotoxic T cells, but also other immune cells in our body as well. So given this wonderful potential for these cells to detect and remove cancer, in recent years they've been used for in therapies, specifically chimeric antigen receptor therapies. These therapies use a synthetic receptor called what we call a CAR that uses an antibody fused to the intercellular signaling domains of a T cell receptor. What this means is that these cells can be engineered to detect certain antigen or certain molecules expressed on cancerous cells, and then activate in a way similar to how they would through their endogenous T cell receptor. And it's been very effective in treating a variety of bloodborne cancers and is being used in clinical trials now for a variety of solid tumors as well. A few limitations of the therapy, though, based on just how it's produced. These therapies take primary T cells from a patient, they expand them in vitro, transduce them with a viral vector encoding the CAR, and then transfuse them back into the patient. Because many of these patients have been undergoing, chemotherapy and various treatments, there's significant variability in the patient-derived T cells. And in some cases, these patients have lymphopenia, which means they don't actually have enough T cells for the therapy. The viral vectors that are used to encode the CAR, they have limited payloads and provide little control over the integration site in the genome, copy number, and this can actually lead to variable in the magnitude of the CAR expression, but also potentially integrating into oncogenes and cause problems later. And then the in vitro expansion process as part of the T cell manufacturing can lead to exhaustion, which limits the effectiveness of these cells once they're transferred back into the body. So I guess a motivating factor for my work is that we can maybe provide an alternative source for stem cells that could be used in CAR T cell therapies and potentially deliver an allogeneic cell product that could be manufactured at scale with defined compositions of CD4 and CD8 helper and cytotoxic T cells. You'd use more targeted gene editing to put the CAR exactly where we want in the genome, and then generate a naive or non-exhausted CAR T cell product. So to do this, I'm replicating a process that occurs in the thymus. This is a little organ that sits behind our sternum and is where all T cells develop in our body throughout our life. This process is initiated when hematopoietic stem and progenitor cells migrate from the bone marrow and into the thymus at this region here. I should say the thymus is composed broadly of two regions, the medulla and the cortex, and these provide a kind of a spatial context for these cells that may migrate through the thymus through these different regions where they're exposed to different signaling molecules that direct the development that specific for whatever stage they're at. So the cells that enter from the bone marrow, they begin to migrate towards the capsule or the outer edge of the thymus cortex. And during this time, they're exposed to Notch ligands. And this is what really initiates and instructs T cell development and is kind of a crucial signal to drive this early development. Cells then reach the outer cortex, and they start to rearrange the T cell receptor genes. And this is what gives them the T cell receptors and their targeted specificity. But making random rearrangements in the genome is actually a bit of a no-no for most cells. So this is a tightly controlled process. These cells that do successfully rearrange the genome, they need to prove that the T cell receptor is functional, that the rearrangement was in frame so that it can actually encode a protein that can be expressed on the cell's surface. After they do that, then they go through another selection process that makes sure that they're not going to react strongly to self-tissue and cause autoimmune diseases. And finally, cells that pass both all of these steps, they will enter into circulation as mature, naive T cells. In vitro, we can replicate some of these processes. Historically, we've used an OP9 DL4 stromal cell line. These are mouse bone marrow stromal cells that ectopically express the Notch ligand DL4, and they work reasonably well. They've been a gold standard for many years for making T cells. However, just the fact that it uses a xenogeneic cell product and animal serum means it's not really suitable for clinical applications. So my colleague Shreya Shukla developed this engineered thymic niche that really gets rid of the need for a feeder cell layer. And she did this by incorporating a recombinant DL4 molecule, as well as the cell adhesion molecule VCAM1. And we can coat them onto a cell culture plate and culture cells in media containing a few T cell inducing cytokines. Using this process, she found that there was a particular synergy between DL4 and VCAM1 that enabled the generation of these T cell progenitors. And shown here, these flow cytometry plots, you can see that based on this expression of the surface molecules CD7 and CD5, which we use to identify pro-T cells, it was only when we added DL4 plus VCAM that we were able to generate these cells. Did some screening experiments in order to just improve the output, because as I showed in the last slide there, we were only generating about 10% of our population that were actually these T cell progenitors. And this was after 14 days. So I did a screen where I tested a handful of other soluble cytokines and growth factors, and identified these two molecules, interleukin-3 and TNF-alpha, that really had a synergistic effect and enhanced the production of T cell progenitors. And you can see from these plots here, just after 14 days of differentiation, this is starting with umbilical cord blood hematopoietic progenitor cells. we could enable, with these two molecules in combination, about a 500-fold increase in the total number of pro-T cells. And looking at the purity of these populations, we went from about 10% pro-T cells on day 14 to about 70%. So a very marked improvement in the production of these cells. But T cell progenitors are kind of fairly early stage of along the T cell development spectrum, and we wanted to push this assay to support maturation into other stages that get closer to these mature naive T cells. So I developed a sequential factorial experiment design where I could measure the responses of different soluble cytokines that we're using in our cell culture media, and really understand dose responses to these molecules and how they change over time. So this diagram here just shows sort of the composition of the factorial experiments, testing different molecules at in combination at 5 different levels. The actual experiment design was called a central composite design for those who are interested. But I conducted these experiments over 7-day intervals all the way from day 0 until day 42. And what this did was enabled me to measure response to these cells as they change, as they pass through these different developmental states, mimicking the process of the thymus. I then fit a polynomial dose response model to these dose response experiments, and then used regression and multi-objective optimization of the dose response to optimize for the cells of interest. Based on this result, or based on this, we got these predictions here for the 6 cytokines that we're using in our cell culture media. At each week, we had a predicted optimum concentration. And you can see in the plot on the left here, in the blue, that it's a little bit noisy, but shows a general changes in responses. And some in particular, like interleukin-3, which was one of the hits from my screen. It seems to benefit cells, kind of in the earlier week, and the same with TNF-alpha, whereas other molecules such as IL-7 maybe aren't as important in the beginning, but are more important later. And this is consistent with what we would expect from T cell development. Then I used some averaging techniques to kind of smooth these responses and predict a 3-stage protocol where we have particular concentrations that we're using from day 0 to 7, and then another set of concentrations from 7 to 21, and 21 to 42. So using the same 6 cytokines throughout the entire time, but just changing the concentrations of them at these different time points. And then testing these predicted optimum conditions against control, where we just kept the same cytokine concentrations throughout the whole 42 days. And you can see here that we very marked, very significant different responses in the total fold expansion of cells, where our control and our optimum are actually pretty similar for the first 14 days, but then diverge, whereas the control cells seem to decrease in number. We can maintain reasonably high fold expansion throughout the 42 days. And then if you look on day 42 at the number of cells that are expressing a T cell receptor, so these are kind of more mature T cells at this point, and we've gotten 3 orders of magnitude greater cells out of our optimum conditions. And then looking at the phenotype of these cells via flow cytometry, we can see that these cells are express a T cell receptor along with the CD3 molecule, this population here. And within that fraction, they express CD4 and CD8 predominantly. So these are not quite at the final maturation stage where there would be a CD4 or CD8 single positive T cell, but are just a stage before. But significant differences in the total numbers of cells generated in our optimum versus our control. And then if we culture these cells for 7 more days, we can actually push them to mature into a predominantly a CD8 single positive or cytotoxic T cell phenotype. And then examining these populations, they express molecules like CD27 and CD45RA, which are indicative that these are a naive mature T cell. And importantly, they function like a T cell should. We can stimulate them via their TCR, and they can secrete molecules like interferon gamma and TNF-alpha, which are important effector molecules for removing pathogenic cells. So I mean, as you can see here, these assays are generally producing cytotoxic T cells, and actually generating these CD4 helper T cells has been a bit of a challenge for in vitro. Has been a challenge for the field. So while I don't have a lot to talk about today regarding that, we are thinking about new ways to provide signals that these helper T cells need to develop. And I showed in the beginning here that CD4 helper T cells engage with dendritic cells. I know this is also something that they do in the thymus as they're developing as well, and they provide essential signals that kind of provide that they're necessary for their development. So we're using stem cells and differentiating them into dendritic cells as a potential antigen-presenting cell that could be used to help stimulate helper T cell development. And just shown here, we can quickly generate these cells that have a kind of a star-shaped, dendritic cell morphology, and express different molecules like HLA-DR and co-stimulatory molecules that are necessary for stimulating these helper T cells through their T cell receptor. And this work we've done with a variety of stem cell sources, both from umbilical cord hematopoietic stem cells, but we've also used induced pluripotent stem cells that we've differentiated through the blood lineage, and then done similar optimization steps to enable them to develop into T cells as well. So with that, I'd just like to thank everyone for attending today, and give a thanks to some people, particularly my supervisor, Professor Peter Zanstra, his postdocs in the lab, Gail Michaels, Ross Jones, and their collaborators in the Holt lab, the McNagny lab, and then some financial support. So happy to take some questions, and thanks for listening.
Thank you so much, John, for your wonderful talk, and I cannot wait to hear more in future. And now we have a time for a question and answer. If any audience has any question, you can put in the question and answer box. It was a really wonderful research. I have just one question regarding that, because you mentioned that when you just kind of this T cell is produced, also we have to avoid them to have kind of lead to the autoimmune disease. But how about the other way? If you get a stem cell and differentiate them to the T cell, if you wanted to transplant into the patient, how you can just manage to avoid the rejection of that one? Have you also any thought in this aspect or not?
Yeah, I mean, there's a few things we need to watch out for, graft rejection, but also graft versus host disease. So, currently in this assay, the cells are recombining a TCR randomly, and we can't control what they would react to. So we could use some gene editing strategies to give them a T cell receptor that we know has a specificity that we want. Or we could knock out the T cell receptor entirely and just put a CAR in there that will react to, a cancer antigen. Involving graft rejection, especially in an allogeneic context, there's been a lot of work with immune cloaking, knocking out class 1 and class 2 HLA on the donor cells, as well as some other molecules as well to prevent elimination by NK cells and macrophages. Actually, some of the work that was talked about in the first talk, some of the same molecules that are allowing HSC engraftment would be useful for this context as well.
Hey, thank you so much. And thanks, John, for taking your time today to share with us a little bit about your work. And right now, we are just to stick to our time. We encourage the audience, if they want, you can contact John to keep the conversation going. And I wanted to conclude the session right now. I would like to thanks again to all the speakers, Julia, Paraskevi, and also John for their excellent presentation today. I would like to thank everyone in the audience for their attention and all of networks who are partnering on this speaker series to get platform to the rising star in stem cell and regenerative medicine research. And we hope you are feeling inspired because the future is very bright. On behalf of the Stem Cell Network Society and the partner networks from Canada, Australia, Germany, Japan, Belgium, and Korea, thank you all for attending this series and wish you well. have a good day and night for people from a different part of the world.